Welcome back. We are back with an exciting keynote talk. We have today Dr. Tammy Tang with us. I would like to give some brief information about Dr. Tang. He got his bachelor's degree from Shanghai Jiaotong University in biotechnology in 2008. Then he got his PhD in genetics and genomics from Florida University in 2014. And then he had several postdoctoral training and research in various laboratories. Currently, he is now a researcher in Dana-Farber Cancer Institute. And Dr. Tang has an experience in analyzing large-scale genomic and transcriptomic data and pipelining workflows, as well as analyzing single-cell sequencing data. Dr. Tang, can you hear me?
Yes, I can.
It's a great pleasure to have you here, to have your talk in our symposium. We are so excited to hear about it. So I don't want to steal from your presentation time. So stage is yours.
Thank you, Engine. Sorry if I pronounce your name incorrectly. Thank you for the for the invitation and this kind of introduction. So can I share my screen now? Maybe? Can you see my screen?
Yes, right now we can see it.
Is the presenter mode or?
No, it's in normal mode.
Normal mode. Okay, great. Thanks. So I will go ahead and get started.
Okay.
Okay. First of all, thank you for the invitation again. It's 6 a.m. in Boston. I haven't gotten up so early since high school. It is my pleasure to speak today. My name is Min Tang, or you can call me Tommy. So I am now a senior scientist at Dana-Farber Cancer Institute, Harvard's Comprehensive Cancer Center. I am currently leading the bioinformatics effort for a big consortium project related to cancer immunotherapy. And this is my Twitter account. I usually tweet questions, tips, and papers in bioinformatics, and sometimes complaints on bioinformatics. You are welcome to follow me. I also have a blog that I have maintained for several years. I note down the problems I met and how I solved them in the posts. So I was trained in a wet lab during my PhD, and I'm going to talk about reproducible research in genomic data science, sharing tips and experience that I have during my bioinformatics journey. I hope my talk can generate some inspirations among you. So I consider myself as a genomic data scientist, if there is such a term. So I have extensive experience in analyzing sequencing data for genomics, epigenomics, and transcriptomics. I use R primarily for data wrangling and visualization in the tidyverse ecosystem. I use Python for writing snake-make workflows and reformatting data. And I'm a lover for the Unix commands. I see there's a workshop for Unix commands in this conference too, so I highly recommend you to join it. So all the fancy technology may become obsolete, but the Unix skills persist. So I share my experiences of sequencing data analysis in my GitHub repositories, so you can go and read them. Okay, now let's dive into today's topic, reproducible research. So we all know how science should be done. For example, like we make an observation first, we form a hypothesis, we perform the experiment, and then analyze the data, and we report the finding. And lastly, we invite others to reproduce the results. However, we often hear that one cannot replicate a published study. There is a reproducibility crisis. So if if you are an experimental biologist, you may think experimental studies may be hard to reproduce because the experiments were performed maybe under different environments, different people handled the mice, or different reagents were used. And you may think maybe computational studies should be easier to reproduce. However, according to Titus Brown, a bioinformatics professor in UC Davis, most computational research is not reproducible. Here I quote him. I don't know a systematic study, but of papers that I read, approximately 95% failed to include details necessary for replication. So here is an example. There was a scandal in Science that the researcher in Duke faked the data and built the results on a flawed analysis. And Dr. Keith Brackley from MD Anderson Cancer Center downloaded the microarray data and could not reproduce the results claimed in one of the papers. Through really hard and detective investigation, he finally found that the original authors has one row shifted in Excel and got all the gene names wrong. And unfortunately, the clinical trial built upon the results has to be stopped. This speaks to the fact that how important we should make our analysis reproducible. So I put a YouTube video link here, so I highly recommend you watch this video. Dr. Keith is really fun. So by the way, in the end, I will share my slides to you, so you can take a look later. So here's another example. So in this paper published in Science titled "Rearrangements Bursts Generate Canonical Gene Fusions in Bone and Soft Tissue Tumors." And in the method section for detection of gene fusions, here it is how they describe. We detected gene fusions in regions of genomic complexity using an approach that integrates multiple independent fusion algorithms, and that's it. So without further details of the method, one cannot reproduce the results. Right? The method really matters. So why reproducibility is hard? So think about it for several seconds. And I come up with several reasons. The first, no raw data are available. So if you cannot download the data, you cannot reproduce it, right? And second, scripts available upon reasonable request. I know this is a joke made by many. It's not really an actual joke. It was written actually in many of the publications. So they don't share their scripts. Third, lack of method description. And the version of the tools could be different. R, Python, there's so many different bioinformatics tools, and the different versions can give you different results. And it could be due to different machines, whether you are using a Unix machine or a Windows machine. So if it is so hard, should you care about it? But keep in mind, actually you are going to do the same analysis for sure in the future yourself. So either it's going to be a revision of a paper and you have to do some analysis using the same data again. And this is for your own benefit. So how to ensure reproducibility? And I will share the tools that I use at a high level in the following talk. So the first thing is I will talk about Git version control, Jupyter or R notebook for documentation, and containers to deal with different computing environments. I'm sure we all experience this on versioning our Word documents. So we keep adding the version numbers after whatever the final documents, and until it getting really long. And Git is a software to track changes in any set of files, including code, like usually used for coordinating work among programmers collaboratively developing source code during software development. And Git records all the histories of your files, so you can easily revert back to any previous version. And GitHub and GitLab are websites that host your Git repositories. And I highly recommend putting your code for the published paper on GitHub or GitLab so others can replicate your analysis. So if you haven't heard about Git, I recommend you Google it. There are many online tutorials so you can learn. Now let me talk about notebooks. So a wet lab member has a notebook to document his daily experiments. I used to work in a wet lab, and I wrote down what, when, and how I perform all the experiments on a daily basis. Similarly, a computational biologist should have a digital notebook to document all the analysis as well. The Jupyter Notebook is more Python focused, although it supports other kernels such as R and many other languages. So the nice thing about Notebook is that you can put the documentations together with the code. For example, here you can write all the documentations here. And for example, like in the real analysis, you will document how you download the data, where did you download the data, and how did you download the data. And then you put the commands there. And the nice thing is that those commands can be valued inline. So within a single document, you have documentation, the code, and the results from the code. And I use R more often, and I love R Markdown in our notebook. So it is similar with Jupyter Notebook, but it's more R focused. So this is more, an Rstudio interface using R Markdown. So again, if you haven't heard about Markdown, Google it, and you'll find a lot of tutorials on it. So in this talk, I'm just giving you a high level of the tools that are available. You can always Google and learn. And that's exactly how I train myself, like to be a bioinformatician. So most of the time I Google and read and watch videos. So in addition to document your analysis in a notebook, you should automate analysis as much as possible. I really like this cartoon. So on the x-axis here is the task size, and on the y-axis here is the time spent. And there are two ways to approach the problem, the geek way and the non-geek way. So the geek way will does it manually first, and then gets annoyed as the task size increases. Then he writes script to automate it, and then runs script for the following tasks. And the non-geek way will does it manually, and then makes fun of the geek's complicated method. But in the long run, as the task size increases, he loses. So remember, computers are really good at repetitive work. So automation will make your research more reproducible and save your time in the long run. And there's a good side effect of automation, because the best of documentation is automation. If you have automated everything, you can minimize your documentation. Of course, you still need to document whatever this black box is. Like, for example, if you write a function, you still need to document what the function does and what's the input, what's the output. And you should write scripts for everything unless it is not possible. For example, you have to do some manual editing for the raw data. And if you do that, you have to document it. So document, document, and document. Those important things I say 3 times. And there's a tool called mkdocs, this link. It can translate a bunch of plain markdown files into a really nice website. So I find it's a really nice way to document your projects. So here are some tips for automation. So if you have a repetitive simple task, put them in the shell script. So you can call it like whatever myroutine.sh. And each time you can just invoke this single command rather than typing all the commands line by line. And that also minimizes your mistakes and save your time. And for automation, there are some really good old GNU Make. So if you haven't heard about Make, you should also Google it. And there are more recent workflow languages such as SnakeMake, which is Python based, Nextflow, which is drawing a lot of attention and popularity, and Widow, which was developed in Broad Institute and runs on the Terra platform. So those are really popular workflow languages. And because inevitably, bioinformatic analysis will involve a lot of different steps. So you have to input data, transform it to a certain format, feed it into one tool, and then feed it into another tool, right? And the workflow language is perfectly designed for that purpose. So you can kind of automate your analysis in a reproducible way. So I have a link here, and actually there are many other libraries that you can use. So now let's talk about the containers. So Docker containers, why we use Docker containers? So imagine you are working on an analysis in R, and you send your code to a friend, and your friend runs exactly this code on exactly the same data set, but gets a slightly different result. This can have various reasons, such as a different operating system, a different version of an R package, etc. And Docker is trying to solve problems like that. So you might have used a virtual box. So think Docker as a virtual machine that encapsulates all the software, libraries, and the configuration files. So you can ship this virtual computer to your friend, and he can use the same computing environment to do the analysis. And this in part solved the reproducibility issue. I also put 2 links here for some like tutorials and for how to use Docker and the Singularity actually together. So you can read this later. Okay. The number 0 problem of bioinformatics is converting formats of input and output between different tools. And the number 1 problem is then installing tools. I still remember I can spend a full day just trying to install a tool. It can be really challenging because there are so many bioinformatics tools, and many of them have different dependencies on other software. For example, a particular tool is Python 2, and then the other tool is Python 3. How can you do the analysis if it is dependent on both tools, right? And Conda is a packaging management tool for any language. Python, R, Ruby, and many others. So, and one can set up 2 different Conda environments, one for Python 2 and the other for Python 3 to solve the problem that I just mentioned. And biocoda is a life science centered channel in Conda. So the tip is use Conda to manage your computing environment. That will make your life a lot easier. So there are other important untaught skills that I will talk about: naming files, project organization, data organization, and backup plans. Okay. People think naming files is easy. So what are your file names look like? Is it look like here or here? And which file names are better? Right. So you should use file names like this because those file names are more informative, right? You know what are those files, what are inside those files, what's the content of the files. And there are 3 principles for file names. So I took it from Jenny Bryan. I need to give credit to her. So this is the original lecture slides. So the first is machine readable. So do not put special characters and space in the name. So Unix machines really hate spaces. So don't put special characters and space in the name. And second, human readable. So you should be easy to figure out what the heck something is based on its name. And the name should play well with default ordering. So put something numeric first and use the ISO, whatever the standard for dates. So year, month, date. And the left pad other numbers with zeros. So the file names can be sorted easily by the computer. So again, it seems like only the US using this month, date, and year format. So use the year, month, date format for the date. Okay, and you can deliberately use hyphens and underscores to allow recovery of metadata from the file names, for example, here. So underscore used to delimit units of metadata that we want later. So like this chunk, and this chunk. And hyphens can be used to delimit the words, like really long ones. So use hyphen between the words and underscore between the metadata. And those are some examples of awesome file names, right? So you have the date here, the same thing, then underscore, some other metadata, underscore, some other metadata. So if you put dates or numbers in the front, so it can be sorted really easily. So you get a rough idea, when did you write the script, right? And kind of help you to track. And you can easily recover those metadata using scripts, for example, in R, Python, or other languages, as showing here. So one of the good example in life science is the TCGA barcode. So TCGA stands for the Cancer Genome Atlas. If you work in cancer, you probably have heard about that project. So in the TCGA project, they devised a really nice sample barcode system. So it has so each bit of digits, they encode different information. For example, TCGA is the project name, and then you have like 02 is the GSS, I think, is the site. And for example, 01 here is the sample. And it has different meanings. So 01 is a tumor, and 10 is let's say matching normal blood. So there are really nice information encoded in the metadata. So once you get a sample with a name like this, you get a rough idea what this sample is. But is it a good idea to encode metadata to file names? So I asked this question a little bit some time ago, actually in May. And apparently someone has different ideas. So if the so for example, Jeremy said, if the metadata is wrong, then you need to change the file names and change it everywhere it might have been referenced. And Jeff Gentry, who was in Broad Institute, and he's the guy after Terra, seems to agree with Jeremy. So they make a good point. So if your metadata is wrong, and then you have to change your file names. So the best practice is to have a UUID associated with each sample. And one has a separate metadata sheet like this to record all the other metadata. And of course, one can put it into a SQL database, and then it can be easily queried. I highly recommend you to read this paper, Parallel Sequencing Leaves or What Makes Large Sequencing Projects Successful. If your lab or institute is doing a large-scale sequencing study, I highly recommend you to read it. So you will get a lot of insights from the experience of the authors. Now I will start talk a little bit about data organization in spreadsheets. We use spreadsheets so much, and everybody uses a spreadsheet is great, but we just need to use it wisely. So there's a paper called Data Organization in Spreadsheets. It was published in 2017, but it's so relevant to our daily work. So I will recommend you to read this afterwards. So I have a link here, so you can always find it. There are many common mistakes that we are making using a spreadsheet. But if we making small changes to the way that we format our data in spreadsheet can have a great impact on efficiency and reliability when it comes to data cleaning and analysis. So I took some of the slides from this software data carpentry workshop. So the first mistake is that one, we used to have like multiple tables in the same sheet. I'm sure that many of us have a spreadsheet like this. However, that confuses computer. So if you read this spreadsheet into R or Python, the computers can't figure out the relationships between this table and this table. So don't use multiple tables in the same sheet. And also use the correct null values. For example, if one entry or one is not observed, one metric is not observed, you should put either the recommended way is to put NA or leave it as blank. So there are other ways, like people sometimes put 0 there, and which is not good. And even worse, some people put 999, which is even worse. So the best option is leave it as a blank, or use NA is a good option, or use null. So this I think is good for SQL. And we also try to use formatting to convey information. For example, highlighting here, or leave a blank line here to separate the dates. So the solution actually is to create the extra field to encode which data should be included or not included. Because a programming language cannot read it, your highlighting. And this is related to the file naming problem that I mentioned. So you should use really good column names. So the good names here, so you use underscores to separate a bit of a word. And these are the good alternative. You use camel cases and try to avoid using spaces. And we also need to be cautious with Excel, because gene names can get converted into dates in Excel. So in this 2016 paper, they show there are a lot of literatures have problematic gene names because of this conversion problem. And it is still really common. So it is in 2020, there's a CSV files and you download from the consortium project, but then you still get the March 2, September 2, those names. And actually scientists are trying to rename the human genes to stop the Excel from misreading them as dates. The lessons actually have not been learned. So this is a figure from a study from the same actual group, Mark Zeman in Australia. And you will see the number of literatures that is affected by this Excel error has been unchanged or even increased in recent years. And the other reason not to use Excel is that, so in this example, the COVID-19 results were lost because of using Microsoft Excel, because they were using an old version of Excel, and it can only handle about 65,000 rows, rather than the 1 million plus rows like the recent Excel that actually capable of, and many of the data were lost. Okay, now I will switch gear to talk about project organization. And this is a really old paper, was published in 2009, a quick guide to organizing computational biology projects. So usually you have a folder name for your project, then you have subfolders like documentation folder for all the documents like papers. You have a data folder to save all your raw data, and source and bin folder for your scripts, and there is a results folder for your results. And this is my own example of project organization in RStudio using R project. Take advantage of R project if you're using R. So if it's an R project, you will see this .R project file, and within this folder, I roughly follow the pattern that in this paper. So I have a data folder, docs folder, the results folder, and script folder. So I always read the data from the data folder using the scripts in the scripts folder and generate the results, intermediate results or figures in the results folder. And those are the examples of my R markdown files. So as you can see, I kind of name them by dates first and add some of the metadata, so I know what the script is. So in this blog post by Jenny Bryan, so if you have these two lines, set working directory and remove the environment variables, these two lines on the top of your code, she will come and set fire on your computer. Apparently she was joking, but why is that? Because using absolute path is hard for reproducibility. If someone wants to run the same code, but the path is only in the author's computer, you cannot reproduce the results. Instead, always use here to construct the relative path. So here is an R package that's trying to solve this problem. So if you use here, for example, this is how you construct the relative path using here, data, and whatever the file name. So the path will always be relative to the .R project file. And this can benefit your future self as well. So if you bought a new computer and you just copy the whole project folder into it, and the same code will still work if you open the R project. Because here So the same code here will construct a relative path, and it will figure out the absolute path behind the theme. And always, also remember, always keep the data in the data folder untouched. I usually do this chmod user -w in the data folder. So to revoke users' permission. So you cannot edit or delete the files in the raw data file folder. And finally, I think there are really two really good papers to read after this talk. So one is good enough practice in scientific computing and the best practice for scientific computing. So both are written by the people in the software or data carpentry community. So I highly recommend you to read it afterwards. And there are more readings that you can do. So what they forget to teach you about R. So if you are into R, and there's a package called R environment, so R package to bring the project local R dependencies management to your projects. There is even a really long article to talk about the reproducible data analysis workflow with R markdown, Git, Make, and Docker, many of the things that I've talked in this presentation. Now, of course, I also have a long list of links and tutorials for how to automate your workflow and how to do reproducible research. So the link is here. And this is my talk, and I started with as a web biologist, and this was the first book I used, Practical Computing for Biologists, to start learning Python, Unix, and it's a really nice one. And if after you become a little bit more experienced, I highly recommend this other book called Bioinformatics Data Skills. This is really a nice book. It will take you to a next level of bioinformatics skills. So like you can always read and watch videos, but really you should learn by doing. So if you have a project that's involve like computational biology or bioinformatics, that's the best way to learn it. So learn by doing, enjoy it. And I have also a blog post on the books for bioinformatics or data science. So you can also read for more details later. I think that concludes my presentation today. So what questions do you have? I think I am several minutes, maybe roughly 10 minutes before the time. So what question do you have?
Dr. Tang, thank you so much for this nice presentation. I think it was amazing. Actually, it broadened my horizons because, you know, I am, and I'm sure most of people here, we have a tendency to do not be very careful about, you know, file names or versions of the softwares. You know, we kind of consider these things are like small things, but we don't realize how much these small things cost for us. And actually, I've seen some pretty bad examples in the chat box, especially the one that Melike mentioned. So that's why I think it was really important. It was a really important talk. So thank you so much. And I think we have a question in the Q&A session. You can see down at the Q&A session.
Okay, so let me see. About the reproducing projects that involve multiple people with quite different computational skill set. Okay, I have the problem that in my project, 1 or 2 people have the skill set experience to ensure reproducibility, but then most often the code data is shared with colleagues in other labs who do many small analysis. The rest is, yeah, I think that's hard question. So if some of you are really experienced and some of them are not that experienced, yeah, to be honest, I don't have a really good solution for that. I think the way is that you should try to convince the other parties the importance of reproducibility. And if they are not able to, for example, use Git or GitHub to do this collaborative data analysis, you should at least convince them to write down all the details of their analysis, right? and document it. So I think that's my answer. I don't have a good actual answer for that. So I think it's still a hard problem. Yeah. Okay. Any other questions?
We have other questions in the chat box, questions from our panelists.
Questions. Where can I see it? I can't see it. Maybe let me.
Okay.
Okay.
Let me write you again, maybe.
Okay. Okay. Since you have the other question from Fatima, since you have experience for the both sides, wet and dry, would you like to share some insights regarding the wet lab part to prepare bioinformatics analysis, apart from do not use Excel? Okay, so the wet lab part. So I would highly recommend you to consult or communicate with a bioinformatician or a statistician before you carry out your experiments. So you really need to talk to them first. For example, one of the most frequent problem is that you do the experiments ahead of time and without telling them. And then when you get data, then they found there's a batch effect. So you have treatment and your control samples, and you perform the experiment in different days, and or you sequence them in different days. So in the end, you get a completely confounded experiment. So you don't know in the end whether the differences you observe between your control and treatment samples is due to your treatment or due to your different processing date or processing or sequencing date. So that's one thing I would highly recommend you to do that. And of course, you should always document really carefully how you carry out the experiments, right? The treatment, right? The machines, the sequencing runs, if it's possible, just document everything. Yeah.